Well, hi, everyone, and welcome to today's webinar. So today's webinar is the first of a new webinar series that we're hosting here at Pelago Bioscience to go through the different steps of the drug discovery pipeline. And the first out today in this webinar is Dr. Mary Xu. And I'd like to give a brief introduction to her before handing over to her presentation. So Mary has a bachelor's in science in Bachelor's of Science degree in materials chemistry from Peking University in China. She then earned a PhD in bioanalytical chemistry under Professor Michael Fitzgerald at Duke University. There, her graduate work focused on the development and application of mass spec-based stability proteomics methods to investigate protein-drug interactions. Next, she did a postdoc at the Pfizer Groton site, working on hydrogen deuterium exchange mass spec analysis on membrane proteins. Today, she's a member of the proteomics lab at the Pfizer Groton site, and her work mainly includes cell model characterization and drug targeting convolution using proteomics. So before handing over to Mary, I'd like to ask you all to write any questions that might come up during the webinar in the chat. And in the end of today's webinar, we'll have a Q&A session where we'll do our best to answer any questions and hope we can have an interesting discussion. With that, I'd like to hand over to Mary. So take it away.
Oh. Yeah, thank you, Joachim, for the very nice introduction. Hi, everyone. This is Mary. I'm a member of the proteomics team at the Pfizer Groton site. So my job at Pfizer can mainly fall into two different categories. So the first category is that we use regular proteomics to measure protein abundances and also help characterize cell model and tissue model for the project team and for them to optimize their biological assays. And the second aspect of my job is to apply stability proteomics methods, which I will tell you more about later today in the presentation, to help deconvolute the protein targets for the compounds. And so today, what I'm going to share with you is how we adapt two of our stability proteomics approaches into the 1-part 2D format and how we evaluate and compare those methods using a pan-kinase inhibitor, Staurosporine. So as Joachim mentioned, feel free to enter your questions in the chat box, and I will be happy to address them for you later. So here I want to thank Pelago for inviting me over for this presentation, and thank you for attending the webinar today. So unlike target-based drug discovery program, in phenotypic-based drug discovery, we started with a biological system that's the most relevant to our disease, and then we stimulate the biological system with our drug molecule. And then we pick out the hits that can give us our desirable phenotypes without any knowledge of what protein targets are binding to the drug. But knowing the protein targets can really help us elucidate the mechanism of action for the compound and can help us then optimize the drug molecules and help us minimize the side reactions of the drug. So one popular way to help identify the protein targets for the drug is to use mass spec-based chemical proteomics methods, where we usually modify our drug compound with some kind of affinity handle, and then we can pull down our protein targets using the modified drug. However, in such chemical proteomics approaches, we usually need to modify our drug compounds, and such modification can sometimes perturb the original interactions of the drug and can skew our result of the target space. So to overcome such challenges, in recent years, scientists have been developing this stability proteomics approach, where we measure the protein stability by generating a denaturation curve and monitor how the drug perturbs the protein stability by looking at the changes in the denaturation curve. And in such stability proteomics approach, there is no need for us to modify the drug compound, and we can use the drug as is. That's why we call them the label-free methods. And by using such label-free methods, we can maximize the detection of the original interactors of the drug compounds. And this is exactly what I'm going to focus on in today's presentation. So first of all, I'm going to introduce you to two of the stability proteomics methods, SPROX and TPP. And then I will introduce to you how we adapt them to the 1-part 2D format and evaluate with the pan-kinase inhibitor Staurosporine. And then I will dive into some unique features between SPROX versus TPP. And last but not the least, I will walk you through how we stratify the hits and prioritize the hits with the Staurosporine compound. So SPROX stands for stability of proteins from rates of oxidation. And then in the SPROX method, we started with our protein sample, usually a cell lysate. And then we treat our sample with the drug compound of interest. And we aliquot our sample into different tubes containing increasing concentration of a chemical denaturant. So the chemical denaturant that we usually use include guanidine chloride and urea. And then we then oxidize the protein in each sample with hydrogen peroxide for a fixed amount of time. So we tune our reaction conditions, so mostly only the methionine side chains of the proteins are being oxidized by hydrogen peroxide. And we can imagine that as we increase our chemical denaturant concentration, more portions of the protein will be exposed, will be denatured, exposed to solvent, and more methionine side chains can be oxidized. And so as we increase the denaturant concentration, we can observe a decreasing intensity of the non-oxidized methionine peptides, and we can generate a chemical denaturation curve for the methionine peptides that we are able to identify. So in such method, we usually apply this commercially available resin to enrich for our methionine peptides in order to maximize our coverage. And then if the ligand is perturbing the stability of the methionine peptides, of the protein where the methionine peptide is coming from, then we should be able to see a right shift in case of a stabilization or left shift in case of a destabilization. And then for thermal proteome profiling, or TPP, or known to us as CETSA Explore, instead of using a chemical denaturation gradient, we use a temperature gradient to denature our proteins. So basically, if we increase our temperature, more portion of the protein will be aggregated out of the solution. And what we measure is the remaining soluble proteins in the supernatant. And we can generate a thermal denaturation curve based on this behavior. And similar to SPROX, if the drug compound is perturbing the protein stability, we should be able to see a shift in the thermal denaturation curve for the protein. And in this table showing on the bottom right-hand side, I'm summarizing some properties between comparing SPROX versus TPP. So in SPROX, we require the detection of methionine peptides. And because of that, SPROX can actually provide a protein domain level information. And because SPROX use peptide-based quantification. And in contrast, TPP, what TPP measures is soluble proteins and is a protein-based quantification. And then in addition to cell or tissue lysates, we can also apply TPP to live cells or tissue sections or even whole blood samples. And then because TPP involves irreversible thermal aggregation, there is no general relationship between the melting temperature shift and the compound binding affinity. In contrast, SPROX is an equilibration measurement. And if certain conditions are met, we can actually calculate the KD value based on the SPROX curves. And then later in my presentation, I will describe to you how we adapt both SPROX and TPP to the 1-part 2D format and how we evaluate the two methods using in this format and determine the hits with Staurosporine. So we chose Staurosporine in the first place because it is a well-studied pan-kinase inhibitor with a lot of the known literature KD values. And so it can help us better evaluate and compare our results. And in this experiment, we use K562 lysate and use MS to the third quantification for better accuracy. So in this slide, I'm showing you both the curve fitting and 1-part 2D format schematics. And we actually apply both SPROX and TPP in both the curve fitting and the 1-part 2D format. to identify the protein targets for Staurosporine. So on the left-hand side is the schematics for the traditional curve-fitting format. And in this format, we include 2 biological replicates. And we did DMSO versus a high dose of the compound Staurosporine, and then we include 10 denaturant concentrations for each SPROX and TPP. And as the name suggested, in the traditional curve-fitting format, we generate the full denaturation curve for both SPROX and TPP. And we perform curve fitting and determine the shift in the curve, and then come up with a list of hits. In contrast, in 1-part 2D format, we were able to include 3 biological replicates, 5 different doses, including DMSO, and also 10 different denaturant concentration in a single experiment. And below is the schematics of how we are able to achieve that. So for the sample preparation step, the 1-part 2D format actually includes both the denaturant dimension and also the compound dose dimension. And by incorporating the compound dose dimension, we can actually maximize the specificity of hit determination. So after the thermal precipitation step in TPP or the chemical oxidation step in SPROX, instead of generating full denaturation curve, in the 1-part 2D format, we actually pool all the sample together for a given compound dose across all 10 denaturant concentration. So instead of having to measure the full denaturation curve, what we measure in the 1-part 2D format is actually the area under the denaturation curve. And so the final value that we are able to obtain using the 1-part 2D format is the ratio between the area under the compound curve versus the area under the DMSO curve, which we later call the R value. And then if the compound is stabilizing our protein, we should be able to see a dose-dependent stabilization increase of the R value as we increase the compound dose, as I circled out in blue here. And then below, I'm showing you 2 pioneering publications which implement this 1-part 2D format. So on this slide, I'm trying to compare multiple aspects between the 1-part 2D format and the curve fitting format. So as I mentioned earlier, in this 1-part 2D format, we were able to include 5 different doses, 3 biological replicates, and 10 different denaturant concentrations in one experiment, which is 150 samples. Even though that may sound like a lot, but because we are doing the 1-part 2D format, later we're pooling all the samples across 10 different denaturant concentrations for a given dose into a single sample. So in the end, we only have 15 combined samples. And we are able to handle this nicely with one set of TMT Pro 16-plex reagents. And then because we are pooling samples in the end, we are able to get away with using much less protein material per sample. And because of that, this 1-part 2D format is actually more amenable to high throughput, like plate-based format. And then in contrast, in a hypothetical curve fitting scenario, if we want to do exactly the same conditions, we will end up using 15 times of the TMT, 15 sets of the TMT 10-plex reagent, and using 10 times more protein material per sample. And then on the right-hand side, I'm showing you the protein coverage that we are able to achieve using the curve fitting format versus the 1-part 2D format. In the curve fitting format, because we require the proteins or peptides to be quantified across all the replicates, and also they should have good quality curves. So that's really taking a hit on our final number of assay proteins and peptides. So you can see here that by using 1-part 2D, we are actually able to achieve about twice the number of proteins and peptides compared to the curve fitting format for both SPROX and TPP. So one thing I want to mention here is that we are applying a modified SPROX protocol here, and we are able to achieve 3 times the protein coverage compared to previous literature. I think that's mostly attributable to high pH fractionation of the methionine-enriched fraction of the SPROX samples. So in conclusion, by using the 1-part 2D format, we were able to achieve 10 times sample throughput and twice the number of coverage compared to the traditional curve fitting format. And one important parameter we want to optimize for the 1-part 2D format is the denaturation range that we want to choose. So the final readout in the 1-part 2D format, so the parameter that we measure in the 1-part 2D format is the R value, which is the ratio between area under the compound curve versus the area under the DMSO denaturation curve. So what we want to do here is that we want to come up with a sweet spot or an optimal denaturation range so we can maximize our R value, which help us to maximize our assay sensitivity. So on the left-hand side, I'm showing you curve schematics. So say if we choose a denaturation range that contains a lot of the pre-transition baseline, our ratio will be compressed. So our R value will actually be smaller than what we wanted. And then if we, in other hand, if we choose a denaturation range that contains a lot of the post-transition baseline, there will be no added value to our R value. And we are also like wasting our resources generating the data points in the post-transition baseline. So the takeaway here is we want to avoid regions with minimal differences between drug and DMSO. And actually after simulation and optimization, we came up with a sweet spot of denaturation range, which is the 5th to the 90th percentile of the DMSO transition midpoints. And below, I'm listing the specific ranges that we use for SPROX and TPP. So to demonstrate, on the right-hand side, I'm showing you a distribution of the R value, simulated R value, based on our curve fitting TPP data. So in this plot, in the red, the black line actually demonstrate the simulated R value distribution for all the kinases that we were able to identify in the TPP experiment if we use the full temperature range. So we can see that the black curve is more centered towards 1, and the R value is like more compressed. In contrast, if we use our sweet spot or optimized denaturation range, we can see the green curve, which is more spread out. And that's exactly what we want. So we can have more extreme R values in some cases. So that can help us with the hit determination. So the takeaway here is that we can maximize our ratios and also our use of resources by focusing on the sweet spot instead of just using the full denaturation range. And then on this slide, I'm going to show you how we determine our hits using the 1-part 2D format. So actually, one advantage of using the 1-part 2D format is that the data analysis is more straightforward than curve fitting format. So basically, what we need to do is to calculate the R value, which is the ratio between the intensity for the Staurosporine versus the intensity for DMSO at a specific compound dose. And then we also need to calculate the mean and standard deviation of all the R values that we were able to generate across the data set. And then the students' t-test p-value comparing DMSO versus Staurosporine. And then we will call a protein or a peptide a hit if they satisfy both the significance criteria and the dose dependency criteria. So for the significance criteria, we require the R value at the highest dose to be at least 2 standard deviations away from the population mean, with a p-value at the highest dose of smaller than 0.05. And then for the dose dependency criteria, we require an increasing trend for the R value for stabilization hits as we increase our compound dose, and vice versa for the destabilization. And we will have a higher tolerance for the plateau region for the dose dependency criteria. So to give you guys an example, I'm showing you the data for PRKCQ in the middle panel. So what you are seeing here are bar plots. So each bar represents the R value that we generate, which is the ratio between the area under the compound curve versus the area under the DMSO denaturation curve for each given dose out of the 5 doses. And then the arrow bars represent the standard error across the 3 biological replicates. And then the dashed line is across 1. So basically, if the compound is doing nothing to the protein stability, we should be able to see our data to be centered around 1 and no significant deviation. But in the case of PRKCQ, which is a known target for staurosporine, we're able to see the significant increase in the R value compared to a ratio of 1. And then we are able to see the dose-dependent increase for the PRKCQ peptide in the SPROX case and for the total protein in the case of TPP. So very nice hit behavior. And then on the right-hand side is a distribution of stabilization versus destabilization hits for both SPROX and TPP. And actually over 80% of our hits are stabilization hits in both SPROX and TPP. And later, I will show you more details how we classify and prioritize our hits. But before that, I want to change gears and then first talk about some unique features between SPROX and TPP. So as I mentioned before, SPROX requires the detection of methionine peptides. However, TPP does not have such limitations. So we're not surprised to see that TPP is able to achieve about 1.5 times the protein coverage compared to SPROX, as you can see the number inside the parenthesis. However, because SPROX is a peptide-based approach, it can actually report on protein domain level information. And I also want to talk a little bit about their unique kinase coverage. So on this slide, I'm trying to show you the protein domain level information SPROX can provide us. So on the upper panel are the methionine peptides that we identified as hits by SPROX, which are coming from the kinase domain for these kinases, which are known to be the targets of staurosporine. And we can see all of them are showing nice dose-dependent hit behavior. In contrast, in the lower panel, I'm showing you the data for the methionine peptides that's from the same protein, but non-kinase domain. So we can see that all of a sudden, they lose the hit behavior and they're not showing any changes across the dose range. And actually 55 out of the 74 SPROX kinase hits were identified by a hit peptide from the kinase domain. So this nicely demonstrates that SPROX can provide us, as a peptide-based approach, can provide us a higher resolution of the protein structural information than the protein-based approaches. And here, I'm showing you actually SPROX can offer a larger assay window in the 1.2D format. So on the left-hand side, I'm showing you the distribution of the R value ratio between SPROX and TPP for all the common kinases that we were able to identify between SPROX and TPP. So we can see that the distribution is skewed towards the right-hand side. So that is saying that in general, SPROX is able to generate a larger R value compared to TPP. And then on the right-hand side, I'm showing you some specific examples. So for IRAC4 and RPS6KB1, I'm showing you the data for both SPROX and TPP. And if we look at the y-axis, we can see that actually SPROX is able to generate a higher R value compared to TPP. And especially for the lowest dose, if we only take a look, focus on the lowest dose, we would have missed these two proteins as hits if we only consider the TPP readout. And then actually such observation is not specific to staurosporine because this report from Professor Michael Fitzgerald group at Duke University actually describes the same observation, but with a different ligand, cyclosporine A. So this might be attributed to some more intrinsic differences between SPROX and TPP techniques rather than any specific ligand. So here on this slide, I'm showing you the distribution for the unique kinase hits from SPROX versus TPP. On the left-hand side is a kinase tree map where I show you the distribution of the 33 unique SPROX kinase hits in blue and also the 29 unique TPP kinase hits in red. So what we see here is pretty interesting. So basically, the unique SPROX kinase hits largely come from the atypical kinase family and the tyrosine kinase family. In contrast, the unique TPP kinase hits are mostly coming from the ChemK, AGC, and STE kinase families. And on the left-hand side, I'm just trying to show you some specific examples. So these two proteins, RIOK2 and ChemK2G, both are known targets of Staurosporine, and we have the known literature KD values of those kinases. So RIOK2 is an atypical kinase. And we can see that by SPROX, we can see a nice hit behavior for this methionine peptide from RIOK2. However, for TPP, the data is not showing any changes across all the denaturant, all the compound doses. And that's not because we cannot generate like a good quality thermal denaturation curve. Actually, after checking back to our curve fitting data, RIOK2 actually has a very good quality thermal denaturation curve with a nice melting temperature of 48 Celsius. And then also in the case of ChemK2G, it has a nice hit behavior measured by TPP. However, if we look at the SPROX data, it's not showing any changes, even though we identify this methionine peptide from the kinase domain of ChemK2G. So the specific explanation to such observation is still not clear to us, and we want to follow up with further investigation. But I want to see here is that what we saw here is purely observational, and this actually demonstrates the complementarity between SPROX and TPP and how we can maximize our protein target information by combining the two techniques. So last but not the least, I want to walk you through how we classify our hits and prioritize our hits with Staurosporine. So we found that one powerful way to classify our hits is by doing the k-means clustering. So k-means clustering is a non-supervised machine learning algorithm which helps us classify the behavior of our data. So what we do here is that for both SPROX and TPP, we first scale our data to be between 0 and 1, and then we perform k-means clustering on the stabilization hits because that's also the vast majority of our hit data. And then actually for both SPROX and TPP, we were able to obtain 4 k-means clusters. And then we can actually observe that from cluster number 1 to cluster number 4, we have a higher apparent affinity for Staurosporine. And in the parentheses here, I'm just noting the number of proteins that's belonging to the specific cluster. And more interestingly, we can see that as we go from the weakest looking cluster to the most potent looking cluster, we actually have more percentage of kinases with known literature KD values. And then the literature KD values is actually decreasing as we go from the cluster number 1 to cluster number 4. So this nicely demonstrates that we can classify our hits into various potency clusters by performing the k-means clustering analysis. And then a common problem which we run into very frequently is that we usually end up with more protein targets than we can follow up with, more protein hits than we can follow up with in terms of the protein target deconvolution project. So we actually think one way to help us prioritize our hits is to use the weight of evidence approach. So we think that if we observe the same protein hits by multiple approaches, that's the protein hits that we want to prioritize. And in the case of Staurosporine, there are 40 common protein hits between SPROX and TPP. And these 40 proteins are the hits that we want to prioritize with multiple pieces of hit evidence. And actually, this set of protein contains 32 kinases, and 7 out of the 8 non-kinases are known to interact with kinases. And one thing I want to note here is that for stability proteomics techniques like SPROX and TPP, we're not only able to identify the direct interactors of the compound, but also indirect interactors due to protein complexes. And then to take one step further, we we then take the overlap between the most potent k-means cluster that we were able to identify in the previous slide from SPROX, overlap with the most potent cluster that we were able to identify for TPP. Then we came up with the 7 kinases, which we call our tier 1 hits. So say if we don't know anything about Staurosporine and in the phenotypic screening project, these 7 kinases are the ones that we want to follow up first. And then on the right-hand side, I'm showing you a table of all the 7 kinases from this tier 1 hit list. And then I also note their literature KD value. So basically, the vast majority of them, actually 6 out of 7 have single-digit nanomolar KD value. And then 6 out of the 7 are identified as a hit by the kinase domain peptide in our SPROX experiment. So again, say we don't know anything about Staurosporine because we know that these peptides are coming from the kinase domain, we can hypothesize that the Staurosporine is an ATP competitive inhibitor instead of an allosteric inhibitor. So to summarize, what we learned is, first of all, the 1-part 2D format, we definitely recommended over the curve fitting format because the 1-part 2D format helps us achieve 10 times higher sample throughput. And also we can use 10 times lower amount of sample materials. And we can assay about twice the number of proteins. And also the data analysis is much more straightforward than the traditional curve fitting format. And then before we do our 1-part 2D experiment, we want to first optimize our denaturation range or come up with a sweet spot to help maximize our assay sensitivity and make better use of our resources. And then TPP and SPROX have their unique features. So TPP can achieve a 1.5 times of protein coverage compared to SPROX. On the other hand, SPROX can provide us with protein domain level information and also larger assay window in terms of the 1-part 2D format. And actually, SPROX and TPP were able to identify similar numbers of kinase hits with Staurosporine in this benchmarking experiment. And then we also show that TPP and SPROX can have unique kinase coverage. So by combining the two techniques, we can maximize our target information. And then we also show that k-means clustering is a powerful approach we can use to classify our hits to different potency clusters. And then last but not the least, we can prioritize our hits by using the weight of evidence approach. So here I want to thank Robert, Graham, Steve, and Matt for their support and helpful discussions. And I also want to thank Mario for the protein domain assignment and also Tracy for providing the K562 cell culture. And then I also want to give my special thanks to my PhD advisor, Professor Michael Fitzgerald, who introduced me to the field of stability proteomics. And last but not the least, actually, our work has been recently published in the ACS Chemical Biology Journal. So feel free to check it out for more details of this work. So that's all for my presentation today, and I'm happy to take any questions you have. Thank you.
Thank you very much, Mary, for that presentation. Really good. I think we already have a question in the chat. So okay. I'll read it out, and everyone else, please, if you have any questions, just write them down in the chat, and I'll read them up here in the webinar. So the first question that come in is from Shanshong, and the question is: could 1-part 2D identify not potent hits, for example, 10 micromolar KD compounds?
Yeah, that's actually a very good question, because usually what we observe by using like TPP or SPROX is that like 1 micromolar is kind of the general kind of the recommendation for the KD values. So if the compound is too weak, then we may or may not be able to detect those. But depending on your compound solubility, if it's say in a lysate-based experiments, we can consider increase like the compound concentration to help with such identification of the weaker binders. But usually it's not recommended.
Okay, thank you very much. We'll see if any other questions come up. And as a very general question from me,
Sure.
Regarding using label-free technologies, such as you've discussed today in your drug discovery efforts, could you maybe mention a bit how you think that provides an additional value in your drug discovery projects?
Oh, yes. Actually, the label-free type of technology can provide a lot of additional value because at Pfizer, we use such stability proteomics or label-free methods to obtain additional information that we are not able to obtain from the chemical proteomics methods. And then by using label-free, we can actually save a lot of resources by making those affinity probes, which actually require some intense medicine chemistry resources. And then we actually at Pfizer, we apply such label-free technology to not only try to deconvolute the protein targets for hit compound from phenotypic screens, but also we are using those techniques to help identify off targets for the target-based drug discovery program. So it's definitely has a lot of potential in our company at this point.
Okay, thank you very much.
Yeah, thank you.
And no other questions have come in yet. So I'm just going to give people a few more seconds to, if they're typing as crazy, to get a question in the chat.
Yeah.
But in the meantime, on behalf of Pelago, so thank you very much, Mary, for today's presentation. And thank you for kicking off our webinar series.
Yep, really appreciate the opportunity. And actually going back to the first question about the affinity, and we also did like a distribution of the KD value for the hits that we identified by the 1.2D method. And we do see that by using 1.2D, we're able to pick up those less potent hits comparing to the traditional curve fitting format. So on a relative sense, I do think the 1.2D has a higher sensitivity in terms of picking up weaker binders.
Okay, that's a good point.
Yeah.
Yeah, I think as no more questions have come in yet, thank you here in response to the question in the chat. Thank you for everyone as well who's taken the time to join the webinar today. We really appreciate it. And I think with that, we will end today's webinar. So thank you again, Mary, and thank you all for joining today.
Thank you. Thank you all for your attention.
These people are scientists, directors, and portfolio managers. This guy is a drug discovery project leader. They have one thing in common. They all failed to check relevant target engagement early in their projects, so they can't see where they're heading. That's risky. Ouch. Let's make sure it doesn't happen in your project.